Hi, I'm Anton Petrov. I'm the project leader for RNA resources at the European Bioinformatics Institute. And today I would like to introduce you to Galaxy workflows for analyzing non-coding RNAs using RNA-seq datasets. At EBI, we have two major databases for non-coding RNA biology. One is called RFAM, and that's the database of non-coding RNA families. Using RFAM, it's very easy to annotate a newly sequenced genomes with non-coding RNAs. And I really encourage you to go to rfam.org and see what RFAM has to offer. But today we'll be focusing mostly on RNA-Central. And that is a database of non-coding RNA sequences. As you probably know, there are lots of different kinds of non-coding RNAs. Some are small, such as microRNAs or piRNAs. Others are slightly longer, such as tRNAs. And then, of course, there are long non-coding RNAs that are very, very long. Now, the information about these RNAs is distributed across dozens of different databases. And until recently, it was really difficult to get a comprehensive view of all the information about non-coding RNAs. And perhaps that's why people were focusing mostly on proteins and not non-coding RNAs when analyzing RNA-seq data. But now there is RNA-Central, a unified place that provides access to 45 different databases and over 30 million different RNA sequences. Now you can find a whole range of useful information in RNA-Central about pretty much any RNA type and over 1 million different organisms. So let's take a look at some scenarios where you would find RNA-Central useful. For example, imagine that you are looking at your RNA-seq data and you see that a certain microRNA is expressed. How can you find out what that means? What's the biological significance of that? One way to do that is to navigate to RNA-Central, find that RNA, and examine all the information that's known about it. So let's take a look at the example microRNA page in RNA-Central. So here are all the links to different databases that describe this microRNA in detail. In this particular case, Malacards contains information linking this microRNA to different diseases in human. There is also the genome browser showing the location of this RNA in genome. There are gene ontology terms that are either automatically assigned or manually curated, as they are in this particular case. And that means that somebody read a paper and connected this specific RNA with specific biological function, which is really great. Last but not least, there are also references and publications that help you get started to find more about the specific RNA. Now, depending on the type of RNA, there could be other annotation, of course, that could be useful for you, such as secondary structure, or perhaps the interactions between the microRNAs and their targets, modifications, conserved motifs, or other things. Let's think about other scenarios. Imagine that you have a region of a genome that you see is expressed and you're curious whether it could be a non-coding RNA or not. So one thing you can do is you can go to RNA-Central and pop your sequence into the sequence search. And when you get the results, it will tell you whether it's similar to any of the millions of sequences already in RNA-Central, and it will show you the secondary structure if it's known, the RFAM classification, and there are lots of ways to explore the results in detail. Moving on. What if you wanted to focus on all non-coding RNAs in one go? How can you do that? Well, you can try exploring the RNA-Central Galaxy workflow. And what it does is that it uses this online platform called Galaxy that enables you to perform complex analyses without installing any software or downloading any data on your machine. So I would like to walk you through this workflow in a second and thank Arina Tagmazian, who helped us develop it while she was visiting the team. Now, we published a description of this workflow in current protocols in bioinformatics, and specifically it's found in support protocol 3. So what does it do? here's a high-level overview of the workflow. It starts by importing your RNA-seq data. In this particular case, it has to do with Drosophila melanogaster. Second, you import the RNA-central annotations for Drosophila. Third step is to connect all of that with the reference genome, once again, of Drosophila melanogaster. And once you have these three critical pieces in place, you can import the workflow that's already predefined for you and connect all these pieces of information, wire them all together, and click run. And depending on the availability of the cloud, sooner or later, you will be able to view the results and see which RNAs are expressed more or less in different conditions, and perhaps draw some conclusions using the RNA-Central database. So I hope this inspires you to try RNA-Central and try using Galaxy. And you can use this example to set up your own workflows and perhaps get some inspiration for how to analyze non-coding RNAs in your specific use cases. So I encourage you to go to RNA-Central and find the Galaxy workflow and try it for yourself. If you have any questions, you can contact RNA-Central team and we'll be happy to hear from you. And I hope you find this useful in your work and analyzing non-coding RNAs and using Galaxy. Thank you.